Last time we ended our discussion on hallmark number 5, which was sustained proliferation, and this would be our last lecture on the hallmarks of cancer, in which we'll discuss the remaining 5 hallmarks. So continuing our discussion from sustained proliferation, we've already seen how cancer cells accumulate mutations and how those mutations make changes in the genome that lead the cancer cells to avoid suppression and continue with the faulty genome. And these cells start dividing rapidly, as we studied in hallmark number 5, that these cells get signals to grow. And when a cell has to grow, they release some sort of factors and metabolites. And now that these cells are growing, they would require energy. So our 6th hallmark deals with altered metabolism. And we are going to discuss how cancer cells fulfill their need for energy when they're rapidly growing. So most of you know that cells get energy in the form of ATP. And there are 2 ways to make ATP. We can make ATP by using oxygen, but if there's no oxygen, a cell can still make ATP and save itself from starvation. We call these processes aerobic and anaerobic metabolism. So usually our normal cells use oxygen to convert glucose into energy. But when oxygen is not present or a cell runs out of it, it converts glucose into lactate and generates ATP. However, the amount of energy generated in the presence of oxygen is more when it is generated without oxygen. Now, cancer cells behave very uniquely in terms that they adapt the regular metabolic activities of a cell and convert that pathway to their own needs. And what they do is, despite having ample amount of oxygen, cancer cells are still going to convert glucose into lactate. Experiments have shown that in tumors or in cancer cells, the amount of uptake of glucose is very, very rapid. So if you want to see which one of the cells are cancerous, one way is to give them radio-labeled glucose, just normal glucose that is labeled with a radioactive dye, so that when a cell consumes that glucose, we can detect it and measure the amount of radiation that a cell contains. The more the cell eats radioactive glucose, the higher the energy concentration we would get, and that's how we can actually measure it. So if you want to study cancer metabolism, we can actually monitor the amount of glucose, and that amount gets excessively high in cancerous cells. So let's say if a cell eats 1 teaspoon of sugar a day, cancerous cells might eat 10 teaspoons a day. And what they do is very interesting, because they're not going to store that energy for themselves. So when they are going to convert that glucose into lactate, the surrounding cells in the tumor microenvironment, and they could be cancerous or they could be non-cancerous, but in either the case, lactate and other metabolites are consumed by other cancerous and non-cancerous cells, which further promote hallmark number 5, that is rapid growth. This altered metabolism, in which cancer cells rapidly eat sugar to produce lactate, is called the Warburg effect, and it was discovered by Otto Warburg. But what you need to know is that cancer cells creating lactate for other cells and then other cells consuming metabolites is not really unique, because this shift in metabolism is both temporal and spatial. Temporal meaning it is time variant, which means that at the same time some tumor cells would be producing lactate from glucose while others would be consuming glucose aerobically. And by spatial, I mean that this phenomena could be spaced out in different sections of the tissue, while in one part the cancer cells are consuming glucose to make lactate, while in another section of the tumor those cells are consuming glucose aerobically. But what you need to know is that this phenomena also occurs in normal physiological function. Let's say, for example, in your muscle cells, when you're working out and you run out of energy, So your muscles instantly shift to anaerobic metabolism and you get a short boost of energy. Let's continue moving towards our 7th hallmark of cancer, and that is really interesting because we're going to talk about how these cancer cells evade your immune system. Your immune system is the avengers of your body, and evading them is doing exactly what Loki would. Cancer cells do that by a number of ways, and we're going to discuss the most common of those 2 ways in which a cancer cell escapes from your immune cells in hallmark number 7, which is avoiding immune destruction. All cells of the human body carry with them a tag for identification at immune checkpoints or gateways. So if a cell runs into your immune cell, it has to show some kind of identification so that the immune cell can recognize it as a part of the body and allow it to continue to pass. These badges, these IDs, could either be cell surface molecules, that is, they are present on the surface of the cell membrane, or they could bind to some molecules on other cells that would help them in recognition. So the molecules that are present on the surface could be categorized in 2 ways. They could either be receptors that could bind to something, or they could be ligands, which are the things that receptors can bind to. And cancer cells can avoid immune destruction by using either a ligand or a receptor. Now, if we talk about ligands, cancer cells express a specific ligand called programmed death ligand, or PDL. There are different types of PDLs, but cancer cells exhibit PDL1 on their surface. Now, PDL1 is also expressed in pregnancy as well as tissue grafts. It is also used by cells of the immune system to avoid autoimmune diseases. When programmed death ligand binds to a receptor on T cells, it stops the T cell from multiplying in order to prevent an immune response. So for cancer cells, the ligand is PDL1, and the receptor that PDL1 binds to is called PD1. But when we talk about receptors, a specific receptor called CD47 binds to the immune cells and stops them from eating cancer cells. It basically gives the macrophages this signal that tells them, do not eat me, I'm friendly. Hallmark 8 talks about one of the most unique features that is associated with cancers, and that is that they promote inflammation. Tumor microenvironments are usually filled with cells of the immune system, both the innate and the adaptive. And this hustle bustle of the immune cells give rise to inflammation in the area. So we can say that tumor microenvironments are hotspots of inflammation. But these environments can be categorized in 2 ways. So as a norm, researchers now refer to tumors as either hot tumors or cold tumors. It's not about the weather in the tissue, but it's more about the kind of immune response that is being generated inside that tumor microenvironment. For example, hot tumors are tumors in which there is an increased amount of inflammatory response, and there is a large number of immune cells that are continuously invading that area and are rapidly dividing and attacking a tissue, which causes an increase in inflammation. Now, I'm not going into details about what causes this inflammation, but all you need to know is that immune cells secrete some particles or some signals that increase the amount of inflammation in the surrounding tissue. On the contrary, cold tumors are tumors where the function of immune cells is suppressed. And in these environments, we see that there's a very slow amount of immune cell function. And this categorization of hotness or coldness of a tumor is a good example to understand what stage of cancer is. If you're talking about hot tumors, they are more likely to spread and metastasize. But if you're talking about cold tumors, they are going to remain dormant for a long time and hence would represent a slow growth of cancer. So how does inflammation help cancer cells? For starters, inflammation basically accelerates the development of tumor. So if you have a lot of immune cells crowding the tumor microenvironment, it's going to cause stress to the body, and the cells surrounding the tumor are going to differentiate and accumulate mutations. However, these mutations are going to help these cancer cells survive, and these mutations then somehow cause changes in the way these cancer cells are spatially arranged in the microenvironment. And what you can see is that these cells become motile, and then they start recruiting blood vessels towards them, and then they start leaching off ingredients from the blood. One of the markers that we are going to study further in our discussion is the EMT, and inflammation is basically another factor that enables the process of EMT. And we are going to discuss it very soon because EMT is the basis for cancer metastasis. You can see in this picture that in cold tumors you don't see any of the radio-labeled immune cells. However, there is another picture in which cells are surrounding, but they're not invading the tumor, while inflamed tumors show a massive amount of immune cells infiltrating the tumor microenvironment.